All right, everybody. Let's try to start, shall we? I apologize for the circus happening, but sometimes that's it's live television. So here we are. So thank you for coming. We are not in the room we said we were going to be in, and hopefully we don't get booted from here. But if we do, we'll roll with it. I'm Jason Heindel. I just want to welcome you to today's seminar with Dr. Andrew Urachko. As you can see, he's an ASM distinguished lecturer. I'll introduce him in a minute. Before we get to that, I did have a couple slides from the branch that some of you have seen before. You have these slides in your talk. I'm going to roll through them if you.
That's no cool. Please do, and I'll just skip through them quickly and do the part of the right after that.
Yeah. So since he's a distinguished lecturer, we wanted to introduce the lecturer program to you. It's a very popular feature of ASM microbiology, and they've been doing it for 60 years, almost as old as our branch, but not quite. We are older than 60 years. 1,000 plus lectures, which is almost the number of lectures we've had. So they've had more lectures, fewer years. We'll see. And it's provided for the branches. So we usually have at least one a year. Money comes from your dues and other sources like the Maximum Foundation. Why ASM? All sorts of good reasons to be involved with American Society for Microbiology. We have a better slide for the branch itself. You can see that there are many options for joining. If you're not already a member, you should join. You can join at the local level. You can join our branch if you're a student, it's free. You can also join at the national level. These are the prices. It's on a calendar year basis. So if you join now, you're good through the end of December. Check out those sites. We, the branch, have three upcoming, these are the next set of upcoming meetings that we're hosting. So on the end of March, March 31st, Robert Potter from CHOP is going to be presenting, and that will be probably in 106. And then at the end of April, Carrie Ann Burnham from Pattern Bioscience will be presenting. And then we have the student chapter organized monthly meeting with surprise guests to be announced, because I don't think we know actually yet. Maybe we do know. You don't have to say anything. Yeah. It'll be in May. And so also the website's being updated. Be ready to receive an email and check out the website. We have branch officer elections upcoming, and you will be receiving an email probably to discuss how you can petition to be one of the elected officers of the branch. So stay tuned for that via your emails. And here we go. I'm going to actually introduce our speaker now. So our speaker is Andrew Urachko. He is an ASMDL distinguished lecturer, currently professor and vice chairman of the Department of Microbiology and Immunology, and the Carol Feist Endowed Chair in Viral Oncology at Louisiana State University Health in Shreveport, Louisiana. And I'm just going to give a real quick background because we were starting a little bit late. He did both his bachelor's degree. Actually, he did both of his bachelor's degrees, 2 bachelor's degrees, and his PhD at University of Virginia Tech.
The biology degree was the same class as you took for biochemistry. So the biology degree was free, but it's still a separate bachelor's class.
Yeah, you take it, right?
I take it.
Boom. So that was at Virginia Tech, and then he went on to University of North Carolina, UNC Chapel Hill, to do his PhD in immunology. And really, he has an excellent career that I'm not going to go through because I would rather have him tell you about his science, because I think that's what we're really here for. So let me, if I can close this, boom, and start this, hide meeting controls, pointer if you'd like it. Here we go.
All right. So thank you. If you have questions, don't hesitate to stop me. I'm happy to stop and talk about it. So what I'm really going to talk about, so most of my career, at least as a faculty member, and really the second postdoc I did was on CMV, but I started out as an immunologist, and I really put this bottom part. This is the title that's on your announcement, but really I started as an immunologist working on monocytes and macrophages when I got to UNC, and then I really wanted to do host pathogens, so I picked up virology. And as we've been learning more about the virus and aspects of entry, we've gotten a lot of cell biology, vesicular transport, and things like that. And I put this up there because I've given a number of these talks with a lot more of the student chapters, saying that where you start out and where you end, you don't know. And if you had asked me when I was a student or a postdoc, if I was going to do cell biology and vesicular transport, I would have said you're crazy. But what I realized is immunology, biochemistry, virology, cell biology, and I have any other one that you guys work on, they're all much more interdisciplinary, more linked than that. So anyway, that was part of sort of what I wanted to get started as we go into it. Oh, come on.
Let me just try that.
Okay. You still got stuff on there.
Oh yeah, this, I don't know where this is coming.
Well, that's okay. Oh, I mean,
Coming from there. I'm afraid to turn it off.
well, we'll go with it, and hopefully it won't be in the middle of anything. You've heard these slides, but one of the things that ASM always asks you about is, what did ASM do for me? Besides, yes, I'm a distinguished lecturer, but for me, my very first professional talk was so I did my undergraduate and did my PhD at Virginia Tech. Well, my very first professional talk was at an ASM in Virginia. And from there, besides the nervousness and getting on, it was the first time I had a chance to stand in front of an audience and talk about it. But really, two of the people I met at that meeting, I ended up interviewing for postdocs. And even though I didn't take the one in North Carolina that I interviewed with, that was at Duke, but I ended up going to UNC partly because I liked that area. And lo and behold, I ended up doing a postdoc there and then a second postdoc there. Currently, I still work for ASM. I was a former branch, I'm in the South Central branch ASM. I was a president 7 or 8 years ago. I'm now currently a treasurer. And part of my role is to give advice, and sort of my turn now to return the favor by giving talks here. I don't know. And we'll go that way. Hopefully, so they asked you to say, what did I do? What did I want to start to do? So when I grew up, when I was a little boy, I wanted to be a marine biologist. So for whatever reason, I like science. At the time, there's a lot of National Geographic. I don't know if you all probably old enough to remember. Yeah, you were, with Jacques Cousteau and all of that. It was fascinating on TV. I mean, in there, exploration of the sea. Let's hope it works. And I actually did get certified as a scuba diver, and that is in fact me. We had a meeting in Australia, I got certified, so I followed part of it, but I didn't go down this route. Somewhere along the line, my grandfather told me about Uncle Francis Urachko, who worked on the polio virus vaccine with Jonas Salk. And I guess somehow that stuck with me and ended up working on it. But the funny thing was, when I was teaching a lecture on vaccine efficacy and positive outcomes, I found this from the University of Pittsburgh. And of all things, Jonas Salk and Francis Urachko. This is in the flyer. It actually says, I don't think I got the extra part. It says in the background is technician Francis Urachko getting stuff ready. So there it was. And somehow that influenced me. And here I am working on viruses and immunology. So what do I work on? I work on human cytomegalovirus. I'll sometimes call it CMV, HCMV, but it is human cytomegalovirus. It is one of the 9 human herpes viruses. Like all of them, they persist for life. For the US and Western Europe, the numbers are about 47%. Somewhere in the US, the numbers are closer, or outside of the US, there's some places almost endemic, closest to 100%. It's put out here. What's interesting about this particular herpes virus, different from the other 8, is it's really spread in nearly all body fluid. So it gives us some clues to ideas of this virus and dissemination and spread. It's going to be a little bit different from EBV, HSV, and the other betas and alphas and gammas. And I know it's hard to see, but that's basically in there as all body fluid. And we're talking saliva, urine, breast milk, it's sexual transmission, et cetera. So there's a lot of fluid to spread it. Why do we care about it? It's the #1 infectious cause of congenital CNS damage in the U.S. and leading cause of congenital deafness. So it's a significant issue. To put those numbers in perspective, about 1 in 150 to 1 in 200 live births in the United States associated with congenital infection. So it's not a trivial number. It's considered one of the torch agents or torches. This is toxoplasma, rubella, cytomegalovirus, herpes, and sometimes they toss in syphilis as things that cross the placental barrier cause problems. Sometimes you'll hear the CMV people call it a silent epidemic because you don't have, like with chickenpox, get a nice little rash. This one, mama gets something, it's hard to sort of know. I already give you the numbers. In some areas of the world, I know it's hard to see that, but behind this, it says that some areas of the world is up to 8%. So you're talking really a significant number. And it's not trivial when you have some of the children. Luckily, most are asymptomatic, but there's a significant number. As I said, there's congenital damage. Deafness is really the most common, but here's obviously two rather sick children with a very common, these petechial rashes. These are little tiny hemorrhages, really. But it's a very classic a symptom of congenital infection, not to mention some of the others, which I'll show you next. But this is really, unfortunately, one of the worst case scenarios. It's microcephaly. So if you ever thought about the brain, probably thinking about yours, you're supposed to eat good food, make your brain healthy. But during development, as the brain expands, it's while the skull expands. If I don't know if you have children, well, technically I've not had three, but my wife did, but I do have three children, but their heads are squishy. There's a little soft spot. It's because as it expands, the skull expands. When you have a catastrophic failure of brain development, which is what happens with CMV, they get a small skull, obviously, but by default, it's a small brain. They have significant issues, and you can just see it's not normal. You get these weird calcified lesions. So these are the ventricles here, sort of looking like a butterfly in the dark colors. These white spots are not supposed to be there. Some of them, this is one of the better looking ones. Some of them will show, I mean, as big as this separation here, giant white calcified lesions, and really that's a significant a problem in children. And some of the issues when we talk about this virus, and this holds today. I know these are a little older numbers, but like them because they really hit the home. So when you think of born with long-term development, here's CMV, fetal alcohol Down syndrome. And again, I know you guys most of you aren't old enough, but in the 60s, one of the most prevalent ones was rubella, congenital rubella syndrome. If you can go back and look at old time magazines, there's a big push for the vaccination, and now you obviously get vaccinated for that, so you have a problem. But you look at its height, it was less than CMV is now. And if you look at when you ask pregnant women what they're concerned about, and again, this is not because they're not concerned about birth defects, but they've never heard of cytomegalovirus. You can see all of them have heard about HIV, Down syndrome, sudden infant death, some of these others. Here's CMV at the last. So it's a knowledge-based issue. And when you think about it for mama, it's getting infected while she's pregnant, somewhere about 2 months, 3 months prior to conception, about 3-4 months post-conception, really late, maybe 6 months is the danger period. After that, the brain is fully formed, and although that you can still have some issues, it's really that early formative period that is a real problem. And just by washing hands can make a big difference. So it's really a knowledge issue, something that's very unfortunate that's not covered in more detail. It's not just children. It's an issue in AIDS patients. I realize with heart therapy and things like that, it's a lot better, but it remains an independent risk factor. This is one of the classic ones, the retina. I don't know. This is what it should look like, a light little red hue. This white is necrotic. The red is hemorrhaging. This is classic CMV retinitis. And again, its numbers are much lower now because of heart therapy for HIV. But nevertheless, it remains an independent predictor still to this day of more negative outcomes. Where we actually have a grant on is studying it in latency, and I'm going to talk more about the entry, which is the other project we have, where it's a real problem in solid organ and bone marrow transplants. And again, you don't have to be a radiologist to look at this, but this is interstitial pneumonia, this foggy colors in the lungs are not supposed to be there. They're supposed to be much darker, almost much more like what would be expected at the top here. And that's a classic, unfortunate consequence of CMV post bone marrow transplant. So it remains really a problem with that. And again, it's hard to see, but all transplants are looked at for CMV serostatus. So you get a solid organ of bone marrow. They know what is the stratified risk factor. In solid organ, it's the donor. In bone marrow, it's the recipient. So if you are seropositive, getting something, that's where you're a high risk. Where if you're the donor organ for a solid organ, it's the virus coming in with the donor. So they look for that because that affects outcome of treatment. A couple others, let's go through quickly. It does like EBV. There's a mono associated with CMV. There's pretty good evidence, at least in cardiovascular disease and certainly in animals, rats and mice. It follows Koch's postulates. They can't do that exact experiment, of course, in humans, but it's certainly linked to a number of different vascular aspects. There's some data, depends on who you talk to. Some are very vibrant about possible link to oncogenesis, and certainly the animal models say Yes, it certainly could be, but I think at this point the jury's still out. And then the last thing is, again, it's hard to see it there. There's some evidence that there's decreased lifespan. So seronegative individuals seem to live a little bit longer than seropositive, only in so much that seropositive people sometimes late in life have cardiovascular episodes that seem to be linked to CMV. So that's where that data comes from. So what we're working on, again, getting at the virus and immunology from my first slide, is CMV is a myelotropic virus. Monocytes and macrophages are really essential. So it's essential for viral spread, viral persistence, and lifelong infection. And like all these herpes viruses, once you get it for life, once you get it for life. You cannot get rid of it. So lifelong persistence becomes a problem. And what we're really looking at is understanding mechanisms of viral infection. We're focused really on the monocyte level and put it in the context of viral disease. So what we're trying to do is say, what if you're the virus, why are you infecting these cells, and what's the benefit of doing it, and what's the outcome, and why would it do that? With the idea of saying, once you know why, you start to learn a little bit more about disease. And really, what from a scientific standpoint, we're understanding the processes required for dissemination. I know it's hard to see there. It's dissemination and persistence. So spread from blood to organ tissue, and then persistence, and hoping we can find clues to really disease, which I think we're doing. I don't think we've found anything that's leading to new drugs, but the ultimate goal is to do stuff like that, because if you don't have the basic science, you can't get to the translation. We talked about that earlier. I don't know if you ever thought about it, but that promoter use for all your plasmids. You ever looked at your plasmid and it says CMV promoter? That is the major promoter to turn on the very first series of genes. And someone at the University of Iowa in the 70s patented that, made $150 million. Remember, this was in the 70s, 80s, 90s, and went off somewhere in, I don't know exactly when it off, but again, basic science of what at the time was just an innocuous virus. Yeah, it had some bad problems with children, but people didn't think about that. Discovered a promoter, turned out to be extremely useful for many, many things. Why do we care about dissemination and persistence? From a virus standpoint, this is essential to viral survival. And what is it? It seeds the organs. Think about it. What does your organ do a virus? It's a portal of viral exit, right? An organ that makes a fluid, salivary gland. What fluid? Saliva. What do viruses do that affect people for kissing, right? I mean, that's what half the herpes viruses do, to spread from human oral contact, kissing. It's just a really good example. So seeding of organs. Now, as I said, this virus is found in more body fluids than just saliva, but nevertheless, that's a good example. Establishment persistence. So once you get dissemination to, in this case, the bone marrow, you allow lifelong infection to come back. And no one has to raise their hand, but if you have herpes simplex, when you have colds, when you are studying for exams, what do you get? You get a cold sore, right? Because it's a stress. This meant the virus gets out, and that's what persistence really is. Yeah, no one has to raise their hand. I teach that lecture to the med students, and there's always somebody raise their hand. I say, hey, you're gonna have to raise your hand. Don't put yourself up there. It's also pathogenesis, right? Because getting into organ systems is exactly what you disease. So in children, crossing the blood-brain barrier is where the disease is in children. You can get, unfortunately, overt organ disease in children, which is really the worst case scenario, which is can be often fatal to the child for that case. But in HIV and others, that's the issue. And this is disease in both immunocompromised. I mentioned HIV, transplant. And if you think of a child inside mama, although she's protected by all the things that we know of in that, but the child itself, if it crosses the placental barrier, it's an immune compromised host. And it also, as I mentioned, is disease and immunocompetent. Yeah. So what I'm going to show you here is sort of our pathway. I know it's a little hard with the stuff there, but I'm going to tell you this to tell you why we study monocytes and macrophages. And I'm going to go through some models and summarize a lot of our data before you show some of the new stuff. So body fluids, epithelial cells. Again, saliva is a common one. It is sexually transmitted agent. A lot of moms actually get it when pregnant from changing diapers in children. It's found in urine. So again, why hand washing fastidiously is a major way to prevent spread. But it infects epithelial cells. Generally, you're going to think of oral epithelial cells, but you can think of genital as well. From there, if you've had I'm sure the lectures on EBV infects epithelial cells and B cells pick it up. There's a lot of common ground between that particular gamma and CMV as a beta, is that monocytes pick it up. The difference is, unlike B cells that start naive and then they become long-lasting memory cells, hold the virus, this one infects monocytes, which are sort of like, quote, the naive B cells, are not long-lived. What happens is the virus affects these cells, forces them to become differentiated macrophage. I'll give you some data on that, where you get organ persistence. And if you think about what is a macrophage, it's a naturally long-lived cell that can live for months, years, maybe even decades. And so it's a great site of persistence. What I'm gonna focus on mostly today is this. We have a second project on the bone marrow latency in the context of organ bone transplant. And then in here, if you think about it, what's a macrophage in the kidney doing, right? It has renal tubular epithelial cells. They're the ones that back up to the ureter. And so the virus is inside the kidney and affects the renal tubular cell. The virus is excreted into the ureter, bladder, urethra, and out as urine. Same idea would be with salivary glands, et cetera, for spread from host to host and getting out in additional body fluids. So what have we done? This is summarizing a lot of work from a lot of my students in the lab. I'm not going to go through all the detail. I want to highlight sort of the top and the bottom. Here are the key features. What we started out when we started was we had hypothesized they were Trojan horses. You all know the story of Troy, and you see this little Cheez-Its one, right? Isn't that the one that's on TV right now? Little kid with the Cheez-It, and the thing goes in and opens up. There's something like that. Trojan horse virus uses monocytes as a Trojan horse to get into an organ system, where then ultimately creates a replication permissive tissue macrophage that establishes this viral persistence. And these are all the many studies over the years showing changes in migration, survival, and differentiation that we did, sort of highlighting that model I already showed you. And sort of in a nutshell, what I'm going to talk about here is a model. We put all our data together, and I'm going to highlight one point down at the bottom, which is going to set some of our newer data up. So if you think of what is a monocyte, obviously an immune cell, it's innate immunity, but it's really like a heavily armed janitor, right? If you have damaged tissue, it does tissue maintenance and repair. That's what it does. It cleans tissue, it fixes problems, but it's also a really, really good immune cell. As an innate immune defender, you give it gamma interferon, it goes bananas. It's really good at killing bacteria and viruses. It makes all sorts of good products. And the virus takes advantage of that by infecting these cells, because what is an inflammatory macrophage? It's monocyte. It wants to get out of the blood and go to organ tissue. So if you're the virus, you want to be picked up and then trick the monocyte to say, hey, dude or dudette, go to an organ, right? And this is what you find. This is a virus found head to toe in basically every organ system, at least under abnormal conditions. Obviously, no one cuts up humans who are just infected. So it's hard to say, but under more abnormal ones, you can find virus absolutely in every organ, to basically every organ tissue. So it's really good at spreading. And what it does is it's favoring this idea. And again, I'm not going to talk about the immunology today, but we've done a lot with transcriptomics and phosphoproteomics and that stuff to clarify what it is. We've got a lot of transcriptional regulators in that. But ultimately, what the goal of the virus is dissemination. Hematologous dissemination, spread from blood to organ tissue, and within that context is to get productive infection in those organ systems instead of persistence. And so that's the deal. So what we've been looking at, and I'm really going to focus quickly on this, but I'm going to talk about some of the newer data right after that. So what are the biological features in monocytes that really allow them to serve as vessels, and then some of the triggers we'll talk about that. So really, and for the shortness of the talk, I can't give all our life's work on that. So I'm going to really focus a little bit more on the triggers. So what we hypothesized when we sort of started this many years ago, and I guess it gets back to what I started as really as a postdoc, when we were looking at integrin signaling in monocytes, and we're saying, integrins bind to matrix components and it stimulates monocytes. And when I went from an immunology lab to a host pathogen lab, I said, isn't a virus like a giant cytokine? And that's exactly what everyone in my labs did. They went and said, then nothing. But really, if you think about it, it's a big, giant ball of proteins. Yes, there's DNA in the middle of the capsid, But on the outside is lots of little proteins that bind to things. So it's like a giant cytokine. And really, this is what I've been doing since I was a postdoc to now, is looking at CMV as a ligand to learn clues about virus and monocytes, or at least aspects of that. So what we were proposing, there's a unique combination of receptor ligand interaction. It's a multivalent ligand for sure. And that there's specific changes in the infected monocytes that promotes viral persistence. So what you have as a virus is not just doing the same thing as everything else does. At one level, yes, it's a ligand, it binds to cells and it's important for survival, but it's really triggering the changes that are making the things that are good for the virus, right? I told you on one of the slides at the bottom, I know it's hard to see, makes a replication competent macrophage. That's its home in organ tissue. It does similar things in the bone marrow. That's its home for persistence. So it has to make, at least in my view, the perfect home. And that's what really we put out. So here's an idea of the virus. Here it is, multivalent ligand. When I started in the lab, they're all like, yeah, it's got DNA and all these things in there. And I'm like, yeah, but this stuff on the outside, it's like a giant ligand. And herpes virologists have goofy naming systems for glycoproteins. Really, they took the genome. It's a giant genome, 240,000 base pairs. And they went left to right and said, what looks like a glycoprotein? GA, GB, GC, GD, GE, GF, blah, 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 blah. Later, as we learn more about it, some are not glycoprotein. So there's no GA. There is a GB. Turns out GB is a really important one. It was originally found as a fusion protein. You ever thought about viruses when they're in a membrane? To get across the membrane, you have to fuse things and pull it apart, right? To allow both the virus, it's in an envelope, it's in a vesicle. So it's a vesicle inside a vesicle, and you have to peel the things apart to get the virus out, right? So that's what it does. It turns out with CMV, and this is a lot of the work we did, is actually a receptor ligand binding protein, binds to EGFR, which is really cool, because when we started, it was great controversy because everyone said it can't bind to EGFR. EGFR is not monocytes. Turns out it's not true. It is on monocytes. It does respond to bona fide EGFR. The other one, and again, even goofier, is it's called the pentamer. As you can imagine, it's 5 glycoproteins, GHGL, again, A, B, C, D, E, F. And then they got tired of doing that because they didn't know what all the glycoproteins were. And if you've ever looked at CMV genome, there's a short region called unique short, US something, a long region called unique long, and just numbered across the thing. Turns out 128, 130, 131 are all part of the pentamer. All right. Pentamer, this is what we've done, binds integrins, GB binds EGFR. I'm not going to cover all the signaling things, and I know it's hard to see, but these things are critical for cellular activation, change in the cell, important for viral entry, which is what a lot of my early work was, trying to study how does the virus get across the membrane, and then elements of productive infection. And really, that's in the end what I'm going to focus a lot on today. So before I get to some more of the data, what is these things and what's the benefit? So why does the virus have to bind to it? Like all these viruses, they have to have a target cell. They have to find ligands that they can bind to. So a hepatitis virus, even though they don't necessarily know all the ligands, it finds things on hepatocytes, right? It's why they obviously affect hepatocytes and not cells in the kidney or GI or et cetera. This is a virus that's very broad tropism, although it's very myelospecific as far as dissemination. But so what does it do? It promotes viral entry, which is if you're a virus, outside bad, inside a cell good, right? I mean, I know that's a silly black and white thing, but that's the reality, right? You're outside a cell, you cannot replicate. You have to be inside the cell to replicate. You have, in the context of a monocyte, I showed you it's in the blood. It's not an organ tissue. The key feature is it cannot replicate in the monocyte in the blood, only can replicate in macrophages in tissue. So you have to activate motility, extravasation, get virus from blood into organ systems. And then the other thing it's going to do, which is, like I said, I'll show you some of the new data we have. I'm promoting some older data and some brand new data on this feature and its productive infection. So what we focus on, we started this, let me take that back. So when I started this work many years ago, we always used clinical isolates. So we were very lucky. We could infect monocytes. Lots of people use lab strains that can infect monocytes. Everyone thought we had something funny. What was going on? Turns out as genetics came on, it turned out that this pentamer, we had clinical isolates. The pentamer is lost sometimes within some of the clinical lessons, 3 or 4 passages in the lab. And what you get is from the pentamer to the trimer. Make sure I have my fingers right. 5 to 3. All viruses have the trimer. It's important. Only clinical isolates have the pentamer. If for anyone, I don't know anyone here working bacteria, you have pathogens in the islands and things like that, but many ways that's what this is. So in lab strains and fibroblasts, you don't need it. You get a mutation in any one of these UL things, all 3 of them are dropped. As soon as one of them is mutated, they come together, the whole complex falls apart, you only get the trimer. Luckily, like I said, we were using clinical isolates. We got an original, my mentor at UNC had the original vaccine strain for the virus. And by good luck for us, that vaccine strain was super stable for the pentamer. I mean, like 50 some passages worth of super stable. So we had passage 10, you could grow that sucker for a long time and it still kept infecting monocytes. Where a lot of these clinical isolates, like I said, with 3 or 4 passages, now with bacterial artificial chromosomes, it's easy. So we focused on this and learned a lot from it. And let me give you some of the data here. So we knew that region, the pentamer, was important based on the fact from some of the clinical, but what really was it doing other than infecting cells? Yes and no. We didn't think it was that simple. So what I'm going to show you is a series of data where whenever you have the pentamer, that's really what the clinical-like isolate is, all right? When you see some of this trimer, it's essentially a mutated virus. This particular one is confusing because it's backwards. This is a virus that was a lab strain that was used for lots and lots of people used 8169. It's here, it's a bad bacterial artificial chromosome. And they always called it wild type because they thought it was a wild type virus. Turned out it's not. It was genetically manipulated, in this case, UL31 was mutated, put it back in. So now truly this is a repaired virus. But I've done this with clinical isolates that we've knocked out the pentamer, or lab strains, we've knocked it back in. This is easy to grow. So hence it's a lot easier in the lab in some cases. So pentamer null virus, lab adapted clinical. And when Maciek did this many years ago, this is at 15 minutes. So if you go longer, it goes up. But what we noticed, if you can look at signaling, this is quantitative. And again, this is an older study from about 10 years ago. SART, classic signaling molecule. It's up, quite a lot in 15 minutes. And I know it doesn't look like much, but it is a kinase. But you can notice in the absence of the pentamer, nothing. Paxillin, it's a regulatory element associated with cytoskeletal manipulation and as well as activation of the cell. Again, much higher. ERK, a MAP kinase, a signaling molecule, high, and even in this case lower. Another MAP kinase, very high, low. So what we learned was, so this thing signals if you have the pentamer, at least in monocytes. If you don't, it doesn't signal. What was cool, and I'm not going to show you, but in fibroblasts is the opposite. It tells you exactly why in fibroblasts and these lab strains, you lost this thing because it was negative to those type of things. But the real thing you did was this. Again, I know this is here, but this is the mutant virus. This is the wild type. What you look at it 1 hour in genomic units or the genome, it's about, in this case, 3 times roughly more efficient at entering. So at 1 hour, there's a lot more virus when you have the pentamer. This is just another strain. We've got a whole bunch of these. We've done it with many, many different strains from different organ systems, from different disease patients. It's all the same thing. You have the pentamer, efficient entry. You don't have the pentamer, not efficient entry. So gave us some clues to probably why it gets rid of it, but also that there's something more going on than just attachment, because that was what the original one. It's an actually entry defect. It's not getting in. When we did, this actually came out just last year, so I guess it's a little bit, not quite a year old, but this was from a paper in PNAS, but we did a phosphoproteomic screen. And again, it's getting back at, signaling, no signaling. And I'll show you this in more detail in just a minute. This is just a short one, but you can see black lines are wild type virus with the pentamer, so not mutated. You get lots of, I know it's hard to see down here, but lots of signaling molecules are turned on. When you lack the pentamer, you can see most things are down. And these arrows are simply ones. And this particular one, we confirmed with phosphoantibodies to verify that, in fact, this screen was legitimate. oh crap, you can't see the one spot's right behind there. But this is what Monchek followed up on, saying, okay, so it's not in there, but is it what really is going on? This is actually a fish analysis. It would look like this, except you can see green spots inside the nucleus. So at 5 days post-infection, you can find virus in the nucleus if you have the pentamer. If you don't have the pentamer, as you can see, nothing. If you do PCR, which this would be the same thing, you can find virus there, genomic virus, nothing there at 5 days. And then if we look at 3 weeks, so appreciate this is the length of time it takes to get viral gene expression in monocytes, 3 weeks. When you do fibroblast, you can see these IE genes. This is the promoter, MIP promoter I told you that made all that money, turns this on within about an hour or two in fibroblast. So it sort of stinks for the poor student doing this because it takes 3 weeks, but nevertheless, you can see it's starting the gene cascade. And if we follow this out, you get early and late, and you'll get mature virus release. So it really tells us this was critical to getting the nucleus, staying in the virus, and ultimately starting productive infection. Yes.
Why does it take so long?
Why does it take so long? I'm going to show you that in just a minute. I paid him $5 today. There's a trafficking defect, or sorry, there's a trafficking that's completely distinct in monocytes and their cell types. It actually takes 3 days to get the nucleus. There's another step that we're not sure of, something silencing the genome when it finally gets in the nucleus. So this gives you a little clue to that question. So this is fibroblast epithelial cells and endothelials all would look the same. This would have to be endothelial cells, human aortic endothelial cells, but we took at each of the time points, we isolated nuclei, looked for presence of our genome. You can see 30 minutes, 1 hour, obviously. There, all the cell types looks like that. It means it gets in, and that's been known. This was the oddity, and actually put us on a path, and very fortunate, got some funding on this. They like this idea that in monocytes, you found nothing. Obviously, I'm not showing you 30 to 8 hours because there's nothing 1 day, nothing 2 days. At 3 days, you start finding virus in there. So there's something really delayed in these cells. I'm going to show you some ideas of trafficking. There's clearly something else going on because it does get in, 3 days, but you don't find gene expression until 3 weeks. So there is another step. We have not looked at that in a lot of detail, but this gave us some clues to really open up what we eventually started doing, which was some of the cell biology, and I'll show you some of that data in just a minute. But what I wanted to do was at least set up a platform to make sense of what the data we're looking at. So this is a model that Bailey had made actually a couple years after we started this, but here's again the virus, pentamer, EGFR, binding to integrin, binding to EGF. The virus activates these pathways. We've done extensive work. It gets in the early endosome in about 15 minutes. You can find it there. If you lack the pentamer, it goes to late endosomes. What doesn't enter efficiently, but what gets in goes to late endosomes and gets degraded by about 1 day. There's absolutely nothing left, which fits with the fish one. I know you couldn't see it, but if with the fish one. What's interesting is if you can take a pentamer virus, treat with an inhibitor SARC, and you can make a pentamer virus look like a pentamer null virus, saying that integrin signaling via SARC is essential for this pathway. We also know that EGF is critical for here. Heather did some work where when we add some small molecule inhibitors after at this point, it'll stall right here. And then you watch it out, it'll start again. So it stalls and starts again by simply having viable EGF. And Victoria in the lab is actually continuing this with the trafficking. But so this is the pathway. It goes from early endosome into the transgolgi. I'll show you some high-res microscopy. Those recycling endosomes. This is where the budding occurs. In about 3 days is when you get nuclear translocation. You ever thought about these viruses that DNA is packed in there really tight? As soon as it finds the pore, it's like popping like a balloon, right? Except for this thing solid, it extrudes that DNA under pressure. And this is really a critical feature because, at least for herpes virus, they follow a mammalian paradigm, meaning you have to have DNA in the nucleus. You replicate using normal and the host cell factors in the nucleus. So basically, retrograde like trafficking pattern, as the student said, it goes backwards. I said, that's true. Backwards from a cell standpoint, forwards from a virus standpoint. Forward virus, backward cell, right? So it's a retrograde transport. Like I said, EGF signaling throughout trafficking is important. You can stall it and it'll go. Hopefully you don't need internet here. I told you about G8, the pentamer and SARC being important, and about 3 days for nuclear translocation. So what sort of data do we have? This is an older one that Jun Hong did, but we have a fluorescent labeled virus so you can track it as it moves. So the virus is in the green, and each of these up here, so this is fluorescent images with the color represented up here. So in this case, UL32 is a viral gene product that's GFP. If you saw the virus as an envelope, there's a capsid around the DNA. UL32 is a protein associated with the capsid. So it's GFP, so you can track the actual capsid with the DNA. And so in essence, what we learned at this point was the DNA and capsid is there in early endosome, red, and of course the yellow is the co-localization. I realize these are older data. I'll show you some prettier pictures as we go on. This is UL32, it's a virus with the trans-Golgi network. Here's UL32 with a recycling endosome. Here's now UL32 with the glycoprotein, so it's now the inside and the outside. And you should see co-localization if the virus is a whole particle. And what was an interesting facet here is when you look at fibroblasts, it buds right here. So the capsid empties right in within 30 minutes, which is why it gets new. This was really surprising to us. And again, as science nerds, the virology side was, it's a whole virus here. It's a whole virus here. It's a whole virus here, right? Because it's sort of fascinating. Nothing is dumped in. If you think about it, one of our ideas is it's immune evasion, right? Don't let anything extra see to a cell that wants to present antigen, right? You're a whole virus. It's got a lot of proteins. It's a big virus. Nothing is out except for what's attached to the glycoproteins in those vesicles. At least that's one of the ways we look at it. So what we can see here is virus is in early endosome 15 minutes. It's in the trans-Golgi at 2 hours. It's in recycling endosomes about a day. And understand, there's lots and lots of data that Jun Hong did. This is just a summary of all the other vesicles we looked at that we didn't find the virus and other time points. Whole virus at 2 hours, whole virus at 1 day, definitely not whole virus at 4 days. And we quantitated it doing 2 different ways. This is the older Leica that we had that did it through a computer. We also did blinded ones where we took the 2 colors and we looked for the same spot so that you were like, is it here and here? And you might lined up. So the computer actually did a really good job, and it's a lot prettier than our looking at spots, but we verified that. But what you can see is, again, I know it's hard, it's 15, 30, 1, 2 in a couple of days. This is percent. In each case, what you're looking at is co-localization with the GFP label on the virus. And so here, GB, I know if nothing's 100%, it's science. That 8% is there, but right around 24 hours, you can see it starts to happen 2 days, 4 days. It starts really losing the stability of the virus stay as a whole particle. You can find the virus is early endosomes. It's about 1 hour. It's completely out of early endosome, but you'll see the next peak. Where are we here? The trans-Golgi starts to come up around that time. As the trans-Golgi starts going down, you can start seeing co-localization with recycling endosomes. And right around here, somewhere in this range, I think 3 days is right in here, is you'll start seeing where the virus is entering the nucleus. So it's in discrete vesicles. Sort of showing this, that we can see, 15 minutes, 30 minutes, 1 hour, excuse me, 45 minutes, 1 day here, 2 hours, 5 days here, then the process. We can start taking these things apart. So what we've done here is ask, okay, so we got these patterns. Was it really important, right? You can show this there, but can you show it's important? That viruses in early endosomes was not a big deal because lots of people shown that. We didn't think that was particularly novel. Being the trans-Golgi, our papilloma virologist said, come on, is it really in the trans-Golgi? Why would your virus go in there? Our virus does that. Your virus can't do it too. It was not that colloquial, but he was a good friend of mine. I think we were sort of having a beer at a Friday afternoon, and he was sort of saying, why would it do it? It makes no sense to me. And so we did a lot of extra work to in essence, to make our science better and prove him wrong. In the end, he goes, I believe you. That virus is inside the TGN. What we did, and the first thing we did was you took a drug agent to it, just basically dispersed the TGN. You can see when you look at DMSO, it's in the TGN. You first see that it doesn't get in the TGN. I know it's hard to see down here, but the net result of this, if it doesn't get in the TGN, it doesn't go anywhere else. It gets stuck at the beginning. There's no step 2, no step 3, no step 4. Really showing the TGN was really important. As it turned out, we tried to knock down RAB11 and some of these RABs, the recycling endosomes. The stability of those proteins is really, really good. And monocytes have a shorter lifespan. And when you try to transfect them, you can't really make a monocyte wait weeks to knock it down because it's no longer a monocyte. So we were not able to do that. But nevertheless, we could block here, and nothing else happened. And that's really what the rest of this data is here. I know it's hard to see. But what gave us further clues, I don't have the movie on this one, but we took, around this time, we got again, it's an older microscope now, but it was a high-res Nikon with SIM. And so you could do reconstructions looking at layers going in. And what you can see is here's a virus outside the sitting by itself. But here you can find the virus is actually inside the TGN. And I know it's hard to see, but we started taking slices and you start to see the green appear. So the virus is actually inside the TGN. Again, why it goes from that to the TGN is still not entirely clear, but it absolutely does. And I have more pictures, but it's actually inside the TGN. So it leaves that vesicle. One of the critical features of that, which I don't have a lot of time to show you about, is some of the syntaxing snares that play a role with the secure transport are in fact important. But one of the things we did do along this line was to try to, when we could find some data to actually show what was important, we were able to find out that luckily these small molecule inhibitors that shut off EGFR. So of course, EGFR is often mutated in certain types of cancers. So there's a lot of drugs out there. We happen to like this one, AG1478. It's not, you can't use this in clinic, but we've done gefitinib and erlotinib, which are both clinically relevant drugs, and they do the same thing. They all knock out the EGFR tyrosine kinase. But what you can look at is if you have, so what I'm showing you is high-res microscope. Each dot represents a virus that's measured relative to the TGN. So poor student Heather counted virus to EGM, and you can see how many viral particles. This is multiple donors, many viral particles. So we were able to start quantitating these changes. And what you can see is you have a normal virus at TGN at 2 hours. It's sitting pretty much close to where the limit of detection is. You get rid of the EGFR signaling, it's not where it's supposed to be. And this is the recycling endosomes. Again, the same thing at now 24 hours. You can find lots of it sitting here. And this really, 0.1 is the limit of detection for these the Nikon SIM. You can see it's not there, but the point being that we can now take a signaling molecule, take a small molecule inhibitor, and show it's not where it's supposed to be. We could, what I'm not going to show you, but we could also wash it out. The cool thing is it gets in about 15 minutes. You wash it out. It's out in about 15, 20 minutes, and it starts up again, which was really a cool finding. And Heather got a very nice paper out of it. So let's get back to this molecule, what we're talking about. How does it actually play a role? So I've showed you there's some distinct trafficking. I mentioned briefly that SARC signaling was critical, but what is actually going on and how did we take this to the next level? So this is what, I know it's a cartoon, but it represents pull downs that Maciek did. All that work represented in a couple little cartoons. This is what the pentamer looks. I know when they drew it, it looks like Mickey Mouse. I get that, but there's the pentamer, GHGL, and this is 128. I don't know which one is which, when a little model there, but it binds to integrins. When you have the trimer, what we noticed is that, this is represented what's going on. GH was binding beta 1. It was parts of the pentamer were binding beta 3 and beta 1. In this case, this one bound both. I don't know what the residues are. We haven't done that. But the signaling is very, very different. And what you get is, if you can see it, positive signaling, CSARC required, negative signaling degradation. There's something with how that engages. And that engagement is important for trafficking and for signaling. We use that benefit to do a phosphoproteomic screen, and that's what This one is, it's from Blue Moon Biosystems. It's an ELISA-based screen. It does 1,318 phosphorylated specific sites. And I'm obviously not showing you all 1,318 here. It is in the paper. They when we got it accepted, they said we want the entire thing. So there's a 35-page supplemental figure with the entirety of all of that data done multiple times. But the key was when you have pentamer signaling, lots of things are up. When you don't, you have it down. I know you've probably been asking this, or maybe you haven't. Why is this one up? The reason that's up is that's the EGFR tyrosine kinase stimulated by GB. So whether you have the pentamer or not, GB still binds EGFR and still signals. I know it's not quite as high, but it still signals. And this actually ends up being a really good control because if GB goes up, we know it's working. Everything else goes down. But the funny thing was, in this particular one, as we did it all, P10 was the highest, most rapid phosphorylated protein in our screen at 15 minutes. And at one level, we were like, okay, does it do anything? Is it important? Okay, I thought the colors looked a little different, but so why might it be important? Why do we focus on it? So we got P10, first one, we obviously looked in the literature, I already knew that. So P10, this is what, for anyone who studies signaling, is that if you PI3 kinase is a kinase, it puts a phosphate on the 3 position of phosphatidylinositols, well, it's PI3 kinase. P10 is the phosphate that takes it off, so it puts it back to the phosphatidylol, the 2, and only in the 4 or 5 position, so it's PIP2. So when we found it, we were like, man, that stinks. It's the highest one. We all know this works. But P10 had a whole nother feature. It's a really cool phosphatase. It takes the position of the 3 takes the 3' position of the phosphatol, inositol, makes it the 4,5 only, but it also plays a role as a protein phosphatase. And it turns out as a protein phosphatase, it controls RABs. RABs control vesicles, vesicular transport. And so this ended up being something that was interesting to us. And so we were hoping when we started this that there was actually something here. And just, sorry, I put the little model in here. Just to remind you, this signals, this does not. So what we did was we knocked out P10. We have a system to knock out, use siRNA in primary monocytes. It works really well. So we can knock out P10, scrambled RNA. We've done all the appropriate controls. What was interesting is that when you block P10, you see effect on entry. You see no effect when you don't have the pentamer. Again, consistent that when you have the pentamer, it signals through SARC, and there's a couple other kinases we've looked at, downstream CK2 and some of these others that we have verified in this paper. I'll share the data that regulates something. And so when you block P10, you have a consequence. We also had a drug that you could add at the same time, so we could rule out that siRNA did something to cell. This drug SF1670 is specific for the enzymatic activity of P10. And you can see as you do a dose response, cells that have the pentamer are inhibited as you get a higher dose. There's really no statistical change, maybe slightly lower, but really not statistically significant when you don't have that. So, I mean, it's not affecting entry. There's something unique about P10. If you have the pentamer, there's blocking entry and affecting entry, and something that's not affecting it when you don't. We did all this in multiple cell types. It only affects monocytes. It doesn't affect epithelial cells, endothelial cells, it doesn't affect fibroblasts. So again, something specific. So we went back to the high-res microscope. I'm looking at early endosomes, so the 30-minute time point. Again, what we're looking at is distance of, in this case, GB to early endosome. Here's your virus, pentamer, virus plus the drug, the mutant virus plus the drug. Each spot representing an individual viral particle counted, measured, represent the EA, and obviously multiple donors done it. You can see right here at the limit of detection, you can find virus where it's supposed to be at 30 minutes. Yeah, the drug right where it's supposed to be. Remember, virus turns P10 off, adding a drug, you can't have an off. Well, I guess theoretically have an off, more off if it was not completely off, but I'm just thinking about that. But technically, you can't have an off, if that makes any sense. When you have virus without the pentamer, you can see it's not I know there's some there, but a large amount of it is not where it's supposed to be. Now look what happens when you block P10. Now you see a good amount of the virus is sitting where it's supposed to be. So we took a pentamer null virus. It's not clinically relevant, at least in the context that it's mutated. And we can almost make it look like a wild type virus by changing one protein, meaning how important is the pentamer to signal to get trafficking correctly where it needs to go. We looked at, in this case, this is the one we added after. So one of our concerns was if you added early prior to infection, you could have some other effects. So we added after infection, so it got in, so you couldn't affect binding or entry. And you see essentially the same thing. Pentamer where it's supposed to be, plus drug in, can't have an off, more off. The mutant virus, I know there's some, a lot not. And now you can see there's a shift. So remember, each of these is measured relative to distance, and obviously all the statistics is done showing there. Was really cool. So we did that. We submitted the paper to PNAS. We got good reviews, but the reviewer said, okay, great. So you recovered it. Is it really recovers and make its way to the nucleus? And like, well, that was a good experiment. And again, I know it's hard with the spots here, but what you're looking at is mock over here. This one here is TB40. So it's a different clinical isolate. It's known to be grumpy virus. It doesn't make its way to the nucleus very well, but it makes it there. This is the repaired virus. It gets in really well, obviously much higher than that. The wild type virus, again, I know you can't see it, but it's essentially zero. Sorry, the virus that doesn't have the trimer only doesn't get in. But now you look at this. When we added the drug, yes, it's not the same as this, but some of it recovers. There's clearly something else, and that was an exciting finding. There's more that the pentamer does out after this process, but we can recover a significant amount of virus. Now in the nucleus, it actually replicates and get virus. Now, by its sense, using a single drug blocks them. Obviously, we're not going to promote viral infection, but we've identified why or what the pentamer is doing in the context of a productive infection, the cell type that's important. I'm almost done here. So what I've shown you, this last bit, virus, pentamer, I know I've done a little bit of this, we don't have a lot of time. This is controlling P10. What does P10 do? It plays a role in the phosphoinositol pathway. It also plays a critical role here. And what we did, and we've shown this, is that RAB7 is a protein that, as you get, when it's phosphorylated, it stays as a early enzyme. When you dephosphorylate it, it allows vesicles maturate. Early enzyme matures into a late endosome. And if you're a virus or a bacteria, you don't want to be in a late endosome in monocytes because nothing good happens when digestive enzymes are dumped in. So by blocking that, you prevent dephosphorylation, and you promote this pathway where you prevent late endosomes. And we did show that in the paper. I'm not sure today. I'm just going to end with, here's our model. We've done some EM, very early work, at least it's consistent. Here's when we did this, the guy came and looked at me and said, what do you think? I said, that's great EM of a monocyte. Where's my virus? And he said, we went and looked back, not all of it, but there's the cool little virus right there. It's sitting right here. It's a little finger-like, is a classic micropenisome, right? But what was cool, and when we started this, we didn't know about the vesicular transport. I did this before to give us some clues. And what we can find at 30 minutes, absolutely virus in little endosomes, 1 hour, which all the other cell types you infect, it's never in a vesicle at 1 hour. So we're excited, like it means something. And then as the rest of the work followed out, we really have a cool aspect of trafficking. Again, I'm excited. This is a cell biology that we've learned a lot about vesicular transport and things that I absolutely, when we started, didn't know a lot about. Other than as an immunologist, late endosomes good if you want to kill bacteria and viruses, lysosomes, all those degradative enzymes, all right here. But if you're a virus, nothing good happens down here. So I'll just end with this. I'll thank my people. And I have one funny little slide at the end about a little joke about Texas. Again, this is most of what I focused on here. The reason we focus on monocytes and productive infection is ultimately these are the cells that go from blood to organ tissue. This is where persistence. So examples, a congenitally infected child can shed virus in urine for months or years. Mama, if she chooses to breastfeed, you can find virus in breast milk for as long as mama chooses to breastfeed. So there's a persistence in the body fluids. Saliva is not known the way EBV, but presumably it's like EBV. Some people shed it weekly, maybe daily, monthly. CMV is probably somewhat similar to that. And in the disease states, this is a lot of what we've been focused on with organ and solid organ bone marrow transplant age cancer patients. So in cancer patients, post chemotherapy, they're immunocompromised, and there are issues with that as well. So here's my lab. Only one I don't think is in there, Jamal. He's a newer student, but that's the whole lab. They're the ones that did a lot of the work, or really did all the work. I sit on my butt and write grants. They do the work and look all smiley at me. Well, I guess I'm there too. So I think it's a Cajun place, so we're eating Cajun food, and obviously the waitress or waiter took pictures of us. These are all the people that did the work over the years. I'm not going to list them all. I've had a lot of people over the time. They've been fantastic. Currently, I have 5 students and a postdoc. They're doing fantastic. I've had high school kids, college kids, graduate students, postdocs, young faculty. I think that's even I should have didn't put her on. My daughter's actually working in the lab right now too. So, and the funding down there. But this is, everyone goes to Texas at some point. If you've ever been to Texas, well, I'm ending with this slide. And my wife gets mad at me because every time I go past the CMV station, I get the giggles and she goes, "Stop it." But I was like, "What? It's not an EBV station, it's CMV station." Here's me, CMV negative, CMV positive line, right? It's commercial motor vehicles. But if you work on CMV, who doesn't want to go to see a CMV station in Texas, right? All right, well, that's all I have to say. I'll be glad to take any questions. I'll try to end with something funny. I appreciate you guys here, and we could go getting started finding a fun little room here. So if you have any more questions, I'm happy to answer them.
We have time for like maybe 1 or 2 questions. If they go back there or one here,
Yeah.
I have a topology question. How, at 15 minutes post-infection, when Jonas is hearing on stuff in the like early end zone, and they're still attacked and all the proteins are still in there, how are they influencing E10 phosphorylation? Is that like a EGFR or TK associated?
Yes. So when we start a lot of the work, most of our ideas are centered on receptor ligand. Truly, that you had the burst and that was it. It was surprising that there was signaling chronic going forward. So the initial P10 is truly receptor ligand. So GB binds EGFR, and you can get the TK phosphorylated in pretty much as early as you can do it. It's hard to do much earlier than 5 minutes really effectively. So 5 minutes is about our earliest time point, but in reality, I would be surprised if it's not within seconds, really. So that ligand binds to GB. The other one, the pentamer, I know, binds to both beta-1 and beta-3, and that's also Src signaling is very quick too. So you get Src signaling, again, most of the time we stop at 15 minutes because it's just not worth it. You get Src signaling at 15 minutes and the EGFR TK phosphorylated in about 15 minutes. And so that is what's driving that. Now, P10, it is in our hands exclusively the pentamer. So if you block GB, there's not an effect on P10. We've done a lot of omics, which I, again, didn't have time to show you. We've done EGFR blockage, integrin blockage, and there are things that are EGFR specific. There are things that are integrin specific and things that are obviously both drive it. P10, the rapid one, is very much a kinase-driven event, downstream receptor ligand via pentamer and integrins. But you can also block some of the other kinases, CK2 and a couple of other ones we looked at, I think CAMK2, and that if you block them, so clearly downstream is Src. When we block Src signaling, we found some of those were turned off. Another question, yeah.
Thanks for the nice talk.
You're welcome.
Your virus, your dimer can infect fibroblasts successfully.
It does, correct.
What's the difference in the trafficking pathway between the fibroblasts and the monocytes, and why can't the pathway used also in the monocytes if it's different?
So in the context of fibroblasts, the virus, all available data seems to indicate it fuses at the cell membrane. Why that is, I mean, I don't know. People ask. I wish I could be little bitty and ask the virus to say, "Dude, what's going on?" or "Do that." I don't know, but it fuses there. In epithelial cells, it does, and endothelial cells, it's an early endosome, but it buds out of here. Now, certainly with a lot of these different vesicles where it buds from, it might be the pH. I mean, because we know, so these glycoproteins that serve as fusion proteins, there's a pre-fusion form before it fuses, then it sort of springs out. And that when it springs out is when it starts taking, I think I showed you the membranes, it peels them apart. And so what triggers that is still an unknown thing. They're still struggling with crystal structures of I mean, that's not my field. It's not the crystal structures of GB and all the herpes virus and pre-fusion, but they have ideas what they look like, because you can sort of think they look like they're spring loaded. My feeling has always been GB in KSHV and CMV are also receptor ligand. The other ones, at least the evidence doesn't seem to suggest they are. So it might mean that they're maybe tighter bound. But to get to your question, I don't know why a fibroblast diffuses here. Maybe because it's a very easy, rapid infection. I mean, if you infect fibroblasts in hour, 2 hours, you get IEs turned on. By 18 hours or so in that time frame, you get viral replication, and you get the first virus 48 hours maybe. And if you really get a high dose, they're starting to lyse by 3 or 4 days. I mean, they just pop. Monocytes do not pop. They can make virus. We've got them in culture for, I think our longest we get it was 16 weeks. Just makes low levels of virus the whole time. From a broad cell biology standpoint, fibroblasts are truly virus lytically infected. They get in, it's out quickly. Monocytes, I think, sit in organs, don't make a lot of virus, so they don't cause a lot of inflammatory disease. And so I get back at the two things that I've tried to talk to you about is the congenital infection. So although they can have horrible organ disease, but ones that don't, that might have serious neurological but don't have organ disease, they can shed virus, I mentioned, for months or years. But there's no observable kidney damage in those children. And then again, the idea is that with mothers and breast milk, there's no mastitis or any other defect in mammary gland and milk production. So the prediction based on that is that those organs where macrophages are at, you don't have a lot of pathology, because the virus is, this is my view, is the virus is sort of had a handshake deal with the host saying, look, I'm going to sit here and make low levels of virus. I'm not going to cause a lot of problems. You don't come send your T cells after me. I'm not going to mess with your organ. And I know it's a sort of a silly way to look at it, but if you're a virus, that's exactly what you want to do, right? A virus that's shed in saliva that necessitates human A and human B being next to each other. You don't want Ebola-like symptoms, because there's no, I mean, I'm not trying to be funny, but there's no interaction under those circumstances when you're sick. Asymptomatic is the ideal scenario if it's a close human-human contact. So I really don't know why these other ones are there. I really think some of the data is probably going to come down to what those vesicles are like, and the outcome that in epithelial cells and endothelial cells and fibroblasts, it's a more rapid infection. And I think in macrophages, you have a number of issues going on, but a big part is its persistence and likely immune evasion, but there could be other things. I mean, I could argue that maybe monocytes or nucleus is just not good. They don't want to get in too early because it sets the wrong tone. You wait days and then weeks, you get where it needs to be. You get this persistence with low organ damage. I don't know, but anyway, that's sort of the ideas here. Any more questions? Oh.
Can you say something about the CMV vaccine? Is there any progress there?
So the CMV vaccine is really turned out to be a rather difficult scenario. And obviously today's climate, politics out of it, it's not good to be doing vaccine work right now. But if I take a step back from that, the problem with CMV is there's a lot of strains. You don't appreciate that, but there's maybe hundreds or thousands of different strains. It's actually a weird virus, and it's a DNA virus very stable genome, you think, but it has a lot of things. It almost seems like mutations are almost like HIV, some of these other RNA virus, which is rather unique for having proofreading enzymes, et cetera. So the original virus I used when I was in Clark Wong's lab was the vaccine strain that was used to test people. So if they gave you the vaccine strain, which was called Town Long, the original one that has the full genome, was not mutated, and then gave you the Town virus, it worked great. It absolutely worked great. You were protecting it. The problem is there's hundreds, if not thousands, of different viruses, and they have not found a virus that will protect against all. They've tried GB because you get good antibodies to GB, so presuming it was an antigen, that was a failure. They tested in primates as well. They've tried the pentamer, the trimer, because again, I know I said the trimer, it's the mutant virus, but you have the trimer and the pentamer if you have the pentamer. If you lack the pentamer, you still have the trimer. Trimer is what allows entry into fibroblasts, so maybe I should explain that too. That might be a difference, that the trimer only maybe influences something different, but it doesn't explain epithelial and endothelial cells where you do still need the pentamer. So they've tried that. They've had no success. They just know there's a lot of viruses. There's a real push to get that. So there is a CMV Foundation that has really been pushing around the country to get more testing. So mama goes in. So I made jokes that CMV seropositive, seronegative, but working at CMV, when my wife was pregnant with baby number 1, we went and got tested. We were both seronegative. Baby number 2, both seronegative. Baby number 3, both seronegative. So we never got it from that, maybe because I was too worried and I always wash my hands. My wife tells you I don't like when the babies pee on you and poop on you. So I'd rather fastidious washing my hands. Maybe others do, but she's like, oh, just it. I was like, no, I don't like them doing it on me. But I was very good. I changed diapers. I'm not saying, I just washed my hands afterwards. So I never got it. I don't know if I'm still sero negative, but anyway, I tested for that. But the reality is there's a lot of different strains. And so it makes it very difficult without knowing what is the correct antigen and what will give protection against everyone's strain. It's very, very difficult. And that's just that. But I guess I was getting to the point is they're trying to do at least clinical testing for that. So a place like Canada does it. I am told that Korea does it. I can't speak for every country. I just know when I went to Korea, gave a talk, it was a postdoc colleague of mine, now the chairman at Seoul at the micro, and he says, oh, no. In Korea, we test everyone. I'm like, okay. But they don't do that in the US, but they're starting to try that because of dollars and cents. So mama's pregnant because of the negative outcomes and the cost. If you can find issues up front early, there are some things like just like education. That was a big thing. Telling women prior to that, just again, wash your hands. Be careful with, these are the things they tell them. Wash your hands. If you already have children, don't share utensils, cups. I do not mean to be dirty. If you're going to continue with your husband, wear a condom. And I know she can't get pregnant anymore, but the point being, if dad has it, you could get it. And so these are all the things that they talk to people about to try to mitigate spread to mama during pregnancy. And like I told you, it's about 3 months prior to conception to somewhere around 6 months post is really the danger period when you can have the most damage, because that's during somewhere in that point is during the neurological development. So yes, the vaccine would be great, but I don't know. There's been big failures at big pharma, and they have a lot of them given up. But I know Moderna's still going at it because they're still trying to get EBV. My wife works in EBV, and they're trying to get EBV vaccines. She's doing some of that stuff, but so far not very successful. Any more? You had a question? No?
Well, I still don't understand why things go so slowly. Did you ever try to compare all these signaling with fibroblasts?
So
because signaling are very fast processes, and I'm a bacteriologist, so infect with bacteria on monocytes, it's seen in a few hours, and you kill, So everything is so slow. There's like pathways which either the signal is very weak or why are things delayed?
Well, the virus is delayed. The monocyte activation is not. So they're highly modal. They're actually more modal than adding LPS or PMA. And we've got, so what happens
The
is
movement of the vesicles. whole
the movement of the
system is very
vesicles
slow.
are slow, but I think it's a virus-driven fact that the cells respond very quickly within minutes. You have NF-kappa B activation, all pro-inflammatory genes are turned on. So the cell is very vibrant and robust as far as being active. It's very much a very classic. I didn't have a lot of time talking about it. It's sort of a M1 bias macrophage from all our omics and other profiling. Although interestingly, a lot of the anti-inflammatory cytokines are turned on, so IL-10 and some of these negative ones. So it's consistent with the idea that the cell itself is pro-inflammatory, but its anti-T and B cell molecules are turned on. I wish I could tell you all that. I think retrograde is not the easiest thing to do in leukocytes. Not a lot of things do retrograde.
Some bacteria do it very well and very fast.
Some do it very well, but retrograde is rather rare for viruses. Not a lot of them go backwards in that sense. But again, like I said, it's backwards in a cell, it's forward for the virus. They're outside going in. I personally favor a lot of this is something to do with, well, two things. One, I think it's immune evasion. You have no real antigen. So the glycoproteins can get processed, but the glycoproteins, if you really look, even though we haven't done it here, but my friend in Germany who's done a lot with the exit, you can see wheels. So there's the envelope and the glycoproteins are bound. So I would be surprised if I drew it, it would look like an old wagon wheel where the integrins and glycoproteins are bound, and there's just nothing that's able to be an antigen under those circumstances. Now, why the some that do get degraded aren't serving as antigens, I don't know about that.
My feeling is there must be something which slows down the process. Some of these interactions will be slowing down.
Oh, I'm sure that this is a tailored event. So EGFR, like I said, if you let here and we add drug at say 15 minutes post-infection, this stalls. So it's an active process. When we started, I would have told you 10 years ago, everything's receptor ligand. I was very biased. And I said, everything happens right up here, nothing inside. But what we learned is this event is very defined for you can still block things days late, 24 hours later, and there's still events. So it is an active process, moderate by virus. So your point is well taken. That's sort of what we've come to the conclusion that this EGFR integrin is pushing this at its own slow, unique pace to make it work. And the fact you can block it here, you can block it here. We haven't done too much at this point at 2 or 3 days, but definitely through this part, there are real defined things that you can still block later. So there's a lot of viral driven processes that are very temporal. It's interesting. And luckily there's lots of questions to ask.
All right, I think we should thank our speaker once more.
Thank you.